This Monday was literally a whole day of writing on my thesis, and it goes very slowly. Alright, quickly putting the PCR samples from last week on gel. Let's do it. And now I'm making gel, by the way, you haven't seen this yet. As you can see, it's too hot. Yeah, a little more. Here you can see the gel red. Make sure to mix it very well. Then just put the wells in and let it solidify. Alright, I'm just back from a genetic engineering meeting of an hour, and I prepared all the DNA samples from the PCR from last week, and I'm gonna put them on gel. I got 3 gels, so this is gonna take a while because I have to load 82 samples. The final one. Let's go, all of the 3 have been loaded. As you can see, 82 samples. So now quickly run it. Then I have to make some media. Then I can go and eat. 120 volts. 40 minutes. Alright. Let's go. Alright, I made 6 bottles of LB media I'm going to use tomorrow. And I'm going to sterilize them in the autoclave. Yeah. Alright, the gels are done. Let's check the result. Moment of truth. Would you look at that? Beautiful. That's a very nice result actually. Alright, then next I'm going to do a gradient PCR. My new primers I made in the computer with the primer design have arrived. And with these primers I'm going to try to get a fragment out of a bacterial genome with a PCR reaction. So let's first make the master mixes for the PCR with the new primers. Alright, I've diluted every primer. Now I can finally make the master mixes with these ingredients to do the PCR. I have 7 different master mixes. The droplet you see here in the little tube is the actual genome of my bacteria. It's completely purified, and when I add these master mix during the PCR reaction, what will happen is that the primers will bind a very specific region in this genome, and with every cycle in the PCR reaction, more and more copies of that specific fragment will form. And I will do this for 7 different regions in the bacterial genome. That's why I have all the different primers. That little amount you see in the tip is all the genome I need. This I add to my master mix. Just like that. And during the heat cycles in the PCR, one specific fragment of the genome will now multiply into billions of copies. It's about 6 AM Wednesday. Let's go to campus like every week. Because I have class from 8:30 until 7 in the evening. Wednesdays are quite long days, and I have to travel for 2 hours to get to my campus. It's in another city.
Everything within the company, within the...
I'm going home. I had 3 classes today. Biomolecular nutrition, interface processes of host-associated microorganisms, and management for engineers. Back in Antwerp, let's go. My Antwerp. Bye.
The next morning.
I have 6 cultures of bacteria. I'm going to do this. I'm already at day 3. So I'm gonna start doing this. The goal is to purify my protein of interest and see if it's present. So here's an example of one bottle. I've put in a little falcon tube because I have to centrifuge it. These are bacteria also containing a plasmid, and plasmids are genes, so they code for something, a protein, my protein of interest, which is an antimicrobial, I think I can say that, yes. Now the goal is to see if they actually produce that protein by doing a Western blot. By the way, we centrifuge to separate the cells from the growth media they were in. Because the cells are way heavier, they form a pellet at the bottom when you spin them very fast. Hang on. As you can see here, we centrifuge to separate the cells which are in that pellet at the bottom from the growth media which is above. Next up, I'm going to remove all of the liquid and resuspend the pellet in a lysis buffer.
Here I'm resuspending the cell pellet in the lysis buffer.
Okay, so these are my 6 cell solutions with 6 different proteins of interest, I hope. And now I'm going to sonicate them with this device to really break open the cells and release the proteins inside the cells in the solution. The aluminium foil is precautionary because I don't know the properties of my protein of interest. It might be photosensitive, so sensitive to light. This is just to prevent denaturation of the protein by light. Let's sonicate the cells. As you can hear, it's pretty loud. So this is going on for 10 minutes. Right, 10 minutes have passed. So we can do the next one. Some ethanol. Okay, while my cells are sonicating, I'm going on with the experiment of Tuesday. So this is a PCR reaction. They are finished, and I'm going to prepare these samples for gel electrophoresis with some loading dye. That's a blue coloration, by the way. It's not from the PCR, it's from the loading dye, because the PCR solution is completely clear. And these should contain my new DNA fragments from the genome of the bacteria, which I'm going to check now. And now I only have to add the DNA. The gel electrophoresis samples are done, so they're ready to be loaded on the gel. But first I have to make the gel, so I'm going to do that now. Meanwhile, the final sonication of 10 minutes is happening, and after that I'm going to eat quickly because it's already 1 PM. I've been here since 9 and I don't eat in the morning, so I'm hungry. Alright, all 6 of them have sonicated for 10 minutes, so we're about 1 hour further, a little more. Now I have to centrifuge them for 25 minutes to separate cell waste from proteins, so I can eat while I do this, finally.
A few moments later.
The gels have solidified. Let's load them with my samples.
More moments later.
Every single well is loaded with my sample and ready to go.
Back to the other experiment, I'm filter sterilizing the supernatant so I obtain a protein solution that the bacteria produce, including my protein of interest, the antimicrobial. Filter sterilizing basically means pushing the solution through very small holes only molecules can go through, so large debris and contaminants are removed.
This is now my filtered protein solution. And this is just some cell waste. These are my filtered protein solutions. I now want to check for one specific protein, my protein of interest that the plasmids should have produced. For that I'm going to do an SDS-PAGE and Western blot, but that's for next week actually. Because the antibodies for the Western blot have not arrived yet, so we have to wait for them for a bit. So for now I'm going to save my filtered protein solution pail in the fridge at 4 degrees. And because I can't do the SDS-PAGE and Western blot now, I'm going to proceed with the cleanup of the PCR reaction mixes that are positive. Midnight, let's go to sleep. Happy Friday, last day in the lab. Back in the clean room, and yesterday in the gel some of the new fragments failed. 3 out of the 7 PCR failed, so it wasn't successful in obtaining the fragments from the bacterial genome, and I have to do those again. So I'm again going to make the master mixes for 3 reactions and then 12 for every one of them. Alright, my master mixes for the PCR reaction are done, but I've added an extra component, a GC enhancer, which might increase the efficiency of the PCR reaction. So let's figure that out. Now that the failed PCR fragments are running again in the PCR, I also went further with the fragments that did work. So I have some fragments from a genome of a bacteria, which are genes. So I have some genes isolated from the genome. And the next step is to put those genes, those DNA fragments, in a plasmid, in a circular DNA vector. We're doing that with the restriction digestion, so it's with enzymes that cut open the plasmid and the fragments, and then we can ligate the fragments inside the plasmid afterwards. But first, the restriction digestion I already prepared. They contain everything, so a buffer, the enzymes that cut it, and the DNA. Lunchtime! Both the gradient PCR of the fragments and the restriction digests are ready. I already prepared all of them to load on the gel. Let's check if anything happened, right? And everything is running. Alright, let's put it under UV light. Fingers crossed. And yes, one of the fragments is very good, this one too, but one of them didn't work yet, but these ones are more important. And my restriction digests, as you can see, all worked. Awesome. This one I can throw away, but this one I have to keep because I need to get the restriction digests out, because I put everything in a gel. Put it aside for now. I've just purified the PCR fragments, and now the final thing for today is to purify the DNA out of the agarose. This is from the restriction digests. I'm just weighing the weight of the little shells now, and then I can purify them.
1 hour later.
Little climbing session. Saturday, catching up on some classes from laboratory animal science because it's a class we have to do on ourselves. And there are a lot of them, 34, and I've only just begun. I have to do like 3 to 4 each week.
A mammoth is lactating, so it's producing milk.
Sunday evening, editing day, just as every week. Oh, there is some lightning. And in the day I had a birthday party. Let's edit this video.